hopping online to join us this afternoon for our latest Horizon Series webinar. This is one of a series of webinars that LRIC has put together over a variety of topics in the livestock industry. My name is Kelly Somerville. I'm the CEO of Livestock Research Innovation Corporation, or LRIC. Just for a little bit of background about myself and LRIC as an organization, maybe why this webinar came together. All of the livestock groups in Ontario are members of LRIC. So we work in this intersection of academia, government, and industry, who all have something to do or touch the livestock industry. And we really work to build networks, pull together research priorities, and drive innovation in livestock wherever and however we have the opportunity. So one of these opportunities came to LRIC via one of our friends at the Food from Thought office. So the Food from Thought office is an organization that we've worked with before on several projects, and it was really how we landed on this as our topic for our latest webinar. Preparing for emerging agri-food pathogens is a key research priority funded by Food from Thought. So since 2017, they've supported researchers to build better surveillance, prevention, and collaboration to protect our agri-food system as we face a rise in biological threats globally, with disease transferring from animals to humans and pathogens contaminating our food supply. This topic is extremely relevant to LRIC members, representing not just the commodity groups themselves, but other members of the value chain. Food from Thought has synthesized the research from their program and engaged interest from stakeholders across the livestock sector to prepare this report on advancing resilience to future pandemics and emerging agri-food threats. The Strengthening Surveillance, Prevention, and Collaboration to Respond to Emerging Pathogens report also shares a call to action to prepare for emerging agri-food pathogens developed through the collaboration between academia, government, and industry. You'll see on your screen some QR codes to access both the Managing Agri-Food Pathogens report and the industry brief that was prepared in collaboration with this report. I'll now invite my colleague, Jean, to introduce our speaker for the afternoon.
I'm pleased to introduce Dr. Zvonimir Poljak, I've had the pleasure to know for a decade or maybe two. He's a professor at the University of Guelph, specializing in epidemiology and animal health, with a focus on infectious disease surveillance, emerging pathogens, and the use of data science in veterinary and public health. Much of his work centers on influenza A viruses in swine and other species, while his lab applies machine learning and data-driven tools to understand transmission dynamics, identify high-risk strains, and inform control strategies. Beyond influenza, he studied other important pathogens like porcine reproductive and respiratory syndrome virus and those linked to antimicrobial resistance, always with an eye towards practical application in the field. A major part of his work involves building interactive disease surveillance dashboards to support real-time decision-making across sectors. While his lab is integrating genotyping, analytical clinical text, or conducting spatial or network analyses, his team's goal is to make complex data actionable. He's also involved in shaping graduate students programs such as health informatics, where he sees growing opportunities for students to blend epidemiology, informatics, and quantitative tools. Zvonimir, the web platform is yours.
Thank you, Jean. I thought that I've known you only for 2 years, but good to be reminded. So I'm going to share my screen and start with my presentation. Yeah, good afternoon, everybody. As Jean already said, veterinary epidemiologist working in the Ontario Veterinary College. And I have to admit that I had a bit of a hard time preparing for this webinar because there's a lot of good stuff that I would like to share. And every dashboard that we developed, and there are 21 dashboards right now, has a story to tell. And there was a lot of decision-making going into the preparation, although the output sometimes looks simple. But there was a lot of complex stuff that was going on beyond that. So I had a hard time taking things out. But hopefully we have a long question and answer session, so we will be able to entertain some of these questions. A couple of introductory remarks, and this is just screenshots of some of the dashboards. I just couldn't resist. I'd like to say that yes, my core work is in the area of swine health, but I like to step across different species into different areas. I work with in human health and livestock health, companion animal health, and wildlife health as well. And each time that I step into these other populations, I bring something back, and I'm hoping that this actually contribute to development of me as a researcher and also as an educator. And that's also often a thought that I share with a lot of students, that they should not be afraid of crossing these disciplinary boundaries, whether this is working in the other populations, or whether this is working and learning new analytical tools, or even working with other types of philosophical frameworks towards exploration of science and implementation of science. You always bring something back that you can apply in your core area. My motivation to start and continue with this work actually has two sides. One side is we thought in the beginning, not only me, but we as a group of researchers thought that this would be useful and it would be interesting. And we also applied for the projects and got funding, but it was also partially personal. And as a young professional, when I finished my veterinary degree, I was in a situation to witness what the trade restrictions that are imposed due to fear of transboundary and foreign animal diseases could do to farming communities. And I think that initial feeling also stayed with me throughout the years, because I was working in different capacities with different animal health emergencies, and they're always uncertain. They bring some sense of loss, and at the same time, they also require some urgent and difficult decision making. And of course, they're always emotional. They need clarity. So with dashboards that we are developing, the idea is that we provide some of this clarity to enable timely and informed decision making in real time. And in my mind, this is a core definition of a surveillance. The definition of surveillance that I really like is that this is ongoing collection, storage, analysis of data, interpretation of results, and communication of this information to those who need to know. So when we are developing dashboards, we had many functionalities in mind, and three of them I will be talking a little bit closer in more details today. One of them is situational awareness. The other one is really looking into detection of novel emergent variants of pathogens, and the third one is contributing to this decision making in the field. So let me start first with one example that I think will resonate with a lot of people who are working in the area of animal health surveillance, and this is awareness, situational awareness during H5N1 incursion in different species. So in March of 2024, there was unprecedented event, unprecedented in many levels, because we had incursion of highly pathogenic avian influenza into dairy cattle in the United States, an event that we didn't really anticipate, because influenza doesn't like to circulate, influenza A type at least, doesn't like to circulate widely in bovine. So not only that there was incursion, but there was also a successful transmission, and a lot of different herds were affected all across the United States. And that also elevated not only animal health fears for animal health community, but also public health as well. Now, the other major event happened a couple of months later, because then on October 30th, we had the first report of incursion of H5N1 in a mixed farm, backyard farm with multiple species in swine. And that also elevated some fear, because once you have strain that is circulating in swine, the conventional wisdom is that sooner or later you can expect something like that in people. And coincidentally, we had a meeting of One Health community that was scheduled to occur only a couple of days later. I think it was November 2nd. It was Friday, I know that for sure. And there was a lot of scientists there who are very knowledgeable about influenza, but they don't have really a focus on surveillance in swine. And although this was not agenda item, it still became one of the main focuses of discussion during this meeting. And there was a lot of questions about the influenza level of surveillance in swine, the level of positivity, the subtypes that are circulating. And I was able to pull up the dashboards and have really up-to-date information about all of these questions that were 24 hours old. So with that, we had high confidence in stating that if we had something like that circulating, the surveillance system is capable of catching any unusual cases, and we would have known about that, and we could say that with great level of confidence. So here my aim is to present you the actual dashboards that where I drew my information from, and here we have 21 dashboard stories. I clicked on influenza A virus in swine. And I was trying here to zoom in into a time period that was relevant for the discussion that happened that day. On the upper panel, you can see in blue the number of submissions on a monthly level. You can also see in cyan number of positive farms. In the middle panel, you can also see the percentage positivity. And in the upper right corner, you will see what is the number of positives and percentage positive. Here you see the demonstration that you can stratify on certain demographic strata and also purposes for testing. We can do smoothing of trends of positivity according to what we consider useful. We can also stratify based on time periods such as week, month, quarter, and year. Next, we can look into specific subtypes. And here in the middle row, you can see multiple lines, and these multiple lines are telling us something about the distribution of major subtypes in swine in influenza. You can also see that there are certain curves that look like outbreak, and I'm going to address that in my later presentation. And if you zoom into any of these pie charts, you can see very detailed distribution and breakdown of demographics where certain subtypes are coming from. We can also look into number of positive farms, and while the number of positive submissions was 50%, the number of positive farms in the same time period was 40%. And again, this is something that I will be addressing in a moment. So this is a demonstration of how we use these dashboards to present a situation to an audience that was knowledgeable about influenza, but they were not specifically knowledgeable about nitty-gritty things, about details of everything that is going on in swine populations. Now, occasionally, I'm going to use example of the same dashboard, but for a different audience. And this audience was mostly interested in swine and specifics of different strains that are emerging in our populations. So we were using exactly the same dashboards, and we were trying to compare that with clinical reports and field reports that were coming in 2023. You can focus on this dashboard on this middle panel, and in this middle panel, you have two periods. So the first period is characterized by these multiple lines that are coming up and down, and they represent different subtypes that are typically circulating in swine. These are H1N1, H3N2, and H1N2. So very similar subtypes that we are seeing in human populations. And sometimes one line of one color is dominating over the other ones, but you don't have a clear and consistent outbreak line. And then in a period that starts in early summer of 2023, we have increase and separation of this one outbreak line. And you can see this in olive green color. And that is a confirmation that we have some new subtype that is circulating. This outbreak curve aligned well with reports of clinical signs that we are seeing in population during that time, and that are characterized by explosive outbreaks of influenza that are typical for situations where we don't have any or very little immunity, pre-existing immunity, and therefore clinical signs are widely expressed. These clinical signs will result in explosive outbreaks of coughing and high fever. They have high morbidity and typically last for a very short period of time. So what we were seeing here is emergence of a new strain that was H3N2 subtype, and this new strain was confirmed through sequence information, and it was new reassortant virus that was coming from seasonal human influenza. It was not reassorting in Ontario, but it was introduced from somewhere else in different jurisdictions, but that was the time when it came to Ontario. Now, I would like to stop here and reflect a little bit on that, because I think what we are seeing in animal surveillance is that oftentimes something that looks like a noisy but stable series, like a flat line on average, but with a lot of noise, at the level of a pathogen, will actually have very different dynamics at the level of discrete genotypes. So something looks stable, but in the background, you have a lot of small outbreaks that are happening all the time, and they represent incursion of different pathogens into this population and a lot of small epidemics. And this is one, I think, typical example of this happening. The reason why I'm mentioning that is that we really need to, not only for influenza, but for a lot of other pathogens, we actually need to have molecular testing or sequencing in order to understand the true dynamics of the underlying pathogen ecology. So that's the first thing. The second thing is that situation like this and characterization of viruses like that is useful because it will also inform some control measures. So one control measure here and for other pathogens is also matching circulating strains with vaccines that are available on the market. So again, this is one example where the same dashboard was used, but for different purposes, understanding the underlying disease dynamics and confirmation or contribution to the identification of novel strains. Now, this dashboard is something else, and this is, again, responding to emerging issues, but not by tracking individual genotypes, but addressing the actual needs of the industry. Avian metapneumovirus in chickens is something that has been identified by our industry partners as an important emerging issue. Once we developed this dashboard, then we were able to actually see that, yeah, they were right. Although some of the testing was happening in the previous years, the level of testing really increased in 2024 and 2025 in chickens, and also level of positivity seemed to be increasing, or at least present at a substantial level. Now, why I like this story, it was because it was driven by the industry requests. This was never in any of our research plans to develop a dashboard for metapneumovirus in chickens, but it started to emerge as a request and a need from the industry, and we were trying to address that. And that's an example of how surveillance infrastructure was reacting organically. We didn't just monitor the disease. We, in a way, contributed to the decision-making, and people in the field in practice we're actually using to understand and hopefully make their decisions, form their decision making on the basis of this information. Now, the next story is about the bovine abortion surveillance, and if I would use one keyword for this dashboard, then I would say it's complex, and we are trying to find the meaning. And again, it was on the request of the industry, but there are a couple of components that contribute to this complex data management in the background. The first one is that we have very variable sample submission. So some people are going to, in order to understand what contributed to abortion in cattle, some people are going to submit placenta, others are going to submit fetus, some will submit both. Maybe somebody else will contribute some other diagnostic material. Then when this arrives to a diagnostic laboratory, you have slightly diverse, similar, but slightly diverse diagnostic pathways among pathologists. And many times this is going to be driven by the client's request. And because of that, you have some inconsistent depth of investigation due to resources or client factors. One thing that I can mention here is that it's relatively easy to work with molecular testing because you know exactly what is positive and what is negative and what is maybe suspicious. But then once you start going into more complex investigations, even pure bacteriology, then this becomes very clear what is positive, but what is negative is becoming a little bit less clear. And then you put on top of that these different diagnostic pathways, then everything becomes a little bit blurry. So we were in a situation where we had to combine multiple testing and data modalities. For example, how do we combine histology reports that are partially text-based and partially standardized, and molecular testing and bacteriological testing, for example, as well. We also do have partially standardized outcomes, which is very valuable, but they are not 100% harmonized. Not because this is the lab thing, but because it's an animal health thing. And then on top of the day, once we thought that we have a good product that can be consistently reproduced, then we figure out that different end users actually wanted to have different summaries. So we created both of them. Now, just to explain what you see on these dashboards here, you can have a very broad overview of different causes of bovine abortion with some colors dominating. And they are displayed as bar charts according to the season, a quota, but we can display that by month and by year. If you display that by year, it's going to be very similar information to what you see on the bottom, and this is a pie chart. If you are really interested in specific cause, then you can select a specific cause from a drop-down menu, and then you can look into this specific cause. I presented here, for example, level of abortions or frequency of abortions that were caused by Listeria, but you can also select some other ones. So we like to believe that there is a high epidemiological value despite these challenges, and that it saves some time to our friends in different government agencies and industries when they're trying to analyze and present this data. Now, another dashboard that I would like to present is actually coming from equids, and this is Eastern equine encephalitis. And this is a slightly different disease because it's the only one that has a very clear seasonality. It's normal because it's a vector-borne, mosquito-borne seasonal infection. It's causing high case fatality rate in unvaccinated horses. Vaccines are available. And it's a very rare zoonosis, but if it happens, then it can be very severe. So people are really interested in when the suspicion is becoming more obvious. So what is the temporality of infection? What is the positivity of these samples? And also, where is the geographical location? And we are trying to provide all of this information in this temporal graphs on the left, where you can see very nice seasonal pattern in both the number of tests that are done and also in the middle, the level of positivity. You can also see geographical distribution for the year of 2024, but I have to provide you with one caveat. And this is, the caveat is that we are using postal codes of veterinary clinics as a proxy for geographical location, and that's not perfect. It would be better if we had the actual location of a farm to be more geographically accurate, but we have to start somewhere because the feedback information from our industry partners is really that this is one of the diseases where geographical location or depicting geographical clusters would be of very high value for the industry. So speaking about quality of the data, what I've seen is that with the dashboards, there are not too many dashboard systems where people are using demographics or where people are using some sort of quality indicators. And we are really big, not only in trying to assess how certain pathogens present, what is the level of positivity or geographical distribution, but also what is the quality of the data that is contributing to such things. So what you see here are 3 epidemiological indicators of the quality. The upper panel is specifically for swine, and this is the presence of history information, because if you want to have, demographic data, because if you want to have stratification by demography and by reasons for submission, then we need to have that information present. If it's not present and if it's not handled appropriately, then this is going to be missing information. We are not going to be able to display that. And I'm happy to report that there is a lot of, over the past probably 8 to 9 years, there's a lot of good quality indicators for this component. The other one that is a little bit more tricky, and this is premises ID. And we are using, swine submissions have a lot of premises IDs, which are unique identifiers. And we are not using these premises IDs to pinpoint into specific locations. We are using this in order for us to be able to calculate whether one submission is coming from the same farm in the same time interval. And the reason for that was, I think, obvious when I was doing presentation on the dashboards for swine influenza. You could see that positivity of submissions was roughly up to date in 2025, was approximately 50%. But if we have summary of positivity of farms, it was 40%. So equating submissions to farms is not the same thing. But in order for us to calculate the farm level positivity, we need to have presence of unique premises identifications at a very high level. And we do have that, but you can see this graph that is going up since 2017, not as a sharp incline, but as a gradual increase. So the quality and the presence of premises IDs and valid premises IDs is increasing, and now it's reaching around 95%. That's not the case in many other sectors, and that's one area that could potentially be improved. It can contribute to the accuracy of geographical locations, although we never have intention to provide, to pinpoint into specific locations, but to use this as information for correct identification of the county level positivity or submissions. Now, with this, I would also like to say a couple of words about what's next. And this is, for us, we want to expand into other zoonotic pathogens that also have production impact. For example, Coxiella and Leptospira. But we also are trying to use some of the new developments in data science and epidemiology to look into developing syndromic surveillance using unstructured data. So this summer we will be working on applying natural language processing for submission text. And one typical example that I like to give is if somebody writes on a submission form, that coffin pigs are scouring in Summer Lane, that means that this text is saying something that something respiratory is going on, something gastrointestinal is going on, and something locomotor is going on. And we can have these lines that will tell us whether these syndromes are changing or new things are emerging. At a syndromic way. Because of the way that we approach the bovine abortions and the issues that we discovered in dealing with this multimodal data, we would like to apply something like a large language models in order for us to enhance the accuracy of the entire classification and an ability to apply that to the new data. And finally, we would also like to leverage viral sequence directly for strain level analysis. And before my concluding remarks, I would like to acknowledge all the sources of funding. Research associate Tatiana Petukova, who is actually contributing to the development of these dashboards and doing fantastic work in that. Co-lead director of Animal Health Laboratory, Maria Spinato. Tanya Rossi from Animal Health Laboratory, who is actually your contact person if you wish to gather access to these dashboards. And a number of other collaborators from Animal Health Laboratory and also Ontario Veterinary College. Funding was provided, but initially from Food from Thought, but also Ontario Animal Health Network, Ontario Agri-Food Innovation Alliance, and NSERC Alliance. And AHL, of course, is our main source of data. Now, some concluding remarks. I started with this definition of surveillance and the idea that this is ongoing, keyword being ongoing, collection, storage, analysis, and interpretation of data. What we are trying to do is to convert this data by this analysis into information. And then we are sharing this information to those who need to know. And those who need to know, they do have knowledge to interpret that in the proper context and to use that for the control purposes, and also wisdom when not to use that information for their own purposes. Right now, we have 21 dashboard stories. They are developed to support functions such as situational awareness and contribute to the novel strain detection and hopefully decision-making. In my opinion, monitoring quality of epidemiological metadata is one of the key components and almost as equally important as monitoring diseases itself. And I think with the new tools that are available for us, we should be looking more closely into how do we integrate and combine this multimodal data forms such as clinical text, genomics, and structured diagnostics for further improvements of our surveillance systems. But one word of caution is that this future that I'm hoping is coming very soon will have to balance innovation with well-designed validation and privacy, which should be fundamentals to build trust. And with that, I would like to finish, and I'm open to any questions.
Wonderful. Thank you so much, Zvonimir, for highlighting your incredible research with the Food from Thought initiative. We do have time for about 10 to 15 minutes of questions. I have a few questions myself, but I do want to prioritize our audience. So we'll start with the first one. We have the dashboards are a great tool, but the amount of information available could be overwhelming for non-academics. How do you recommend harnessing these tools to tell a message without getting lost in the weeds?
Yeah, that's an excellent question. I think I'm a little bit biased because we are starting with such a vast amount of information, and we are trying to narrow down into something that is going to be useful. I think it takes a little bit of time to actually learn what's available and what could potentially be useful. And I mean, I have to admit that when I'm looking at swine dashboards, because when I understand the situation very closely, I'm going to look into very, very detailed amount of information. And sometimes it's not going to be enough. It's not going to be enough, specific enough. But when I'm looking into something that I'm not as familiar with, then, the first two dashboards are going to be sufficient, right? So I think it takes a little bit of familiarizing yourself with these dashboards, how they function, and also what type of information you're seeking and for what purposes. We are trying to provide some additional information about the context. So essentially, every dashboard, and I think that was also recommended by people who were working on COVID dashboards, that there should be some sort of a additional information page that provides the details about how these dashboards are created and what should people be, how should they be interpreting that. We are trying to do that, but we recognize that this could be better. So I'm not sure, but I gave accurate enough and broad enough answer, but this is how I'm using and what we are trying to do apart from just providing the dashboard. We are actually trying to do the contextual information as well.
Absolutely. Thank you, Zvonimir. Next question is, have you been able to access any One Health or human health funding streams, or primarily through livestock calls?
Yeah, so far it's been through livestock calls primarily. I think there's the first one was really food from thought, and that was focused on livestock pathogens. Then after that, we had a couple of ON groups who were interested in specifics. For example, bovine abortion was one of them. But we also had some poultry dashboards developed under that scheme. The latest one was really about zoonotic pathogens, and that was funded through the Ontario Agri-Food Innovation Alliance. But they also have production lens. And then we were able to match that with answer discovery. Again, because it's not human, it's actually zoonotic. We never really applied further for development of to human funding streams. One of the reasons is because we are working with animal health laboratory data. But yeah, I mean, there are other streams of data that are within our reach, but we also need to be careful. What we don't want to. We want to be collegial, and we don't want to utilize the data set that somebody already is using for some other purposes. And that's one thing that I forgot to mention, and this is that we are not using research data. That's our exclusion criteria. So only diagnostic and monitoring data are used in these dashboards. So if somebody is using doing research with animal health laboratory, we are not using that data at all. It's exclusion criteria.
Interesting. Thank you. Next question. There's so many questions coming in. This is great. It can be difficult for researchers to form connections with those in other specialties. How do you recommend your students start to engage researchers in other unfamiliar subject areas to broaden the impact of their research?
Yeah, I mean, yeah, thank you for this question. I think it's really forming the collaboration with people. I think we are getting into the area where a single person cannot address everything that is coming our way from all possible directions. So I think this is the way that I'm approaching this is to form the collaborations on projects where everybody's bringing their expertise, and then really have regular interactions with these people, and also encourage students to spend some time with these people because everybody's busy these days. But if they spend some time in their labs or in their just by interacting with them, then I think this is contributing to this. The next thing is also going to conferences that are not directly related to what we are doing. But sometimes you go to a conference and even if you don't understand 100% of everything that is going to be going on there, you can still gain some new perspectives and see how you can apply some of this. And it's a great networking opportunity as well. So I think just everything that is in your disposal, interaction with people, networking, collaboration on joint projects where everybody is bringing their own perspective. It can be challenging. And sometimes I'm in a situation where I said, I have no clue what you were talking about. You have to simplify that for me. And there are situations when I have to simplify that for the other people. But that's how we all learn.
Yes, absolutely. That's a very common practice for researchers, for sure. Another question, what are the challenges you encountered in integrating technology into surveillance of animal diseases, and how have you gotten over those hurdles, and what remains unsolved?
So one big thing is, I think there are a couple of things. One of them is really some data are relatively easy, right? And I think I already alluded to this, because with molecular typing or molecular testing, it's either positive or it's negative. Sometimes it can be suspicious, but then you can also say, well, we are going to assign that to a certain category. With bacteriology, it's a one level above that, because with bacteriology, you have different reasons for testing. Some people are testing for clinical suspicion, and others are testing, or the entire industries are testing that for monitoring purposes. And then because of that, you have different specimens, but you also have different ways that these specimens are handled in the laboratory. And sometimes you actually don't have that information. So it's really, I think, useful to go to a lab and to understand the entire process. That's, I think, other advice that I would give specifically to students and people who are like maybe research associates. Go to a lab and really understand how this process is working and how these data are generated. From the very detail of how this is submitted to a laboratory, who enters that information, who then enters the result of the positive test, and how this sampling and testing protocol actually looks like. As already mentioned, with pure bacteriology, it's sometimes difficult to say what is negative for someone else. It's quite clear what is positive, but what is negative is you really have to have some sort of a thinking about that, what you are going to call negative. Generally, I think what we are lacking in veterinary medicine is sufficient application of standards. In human medicine, this is a very common thing, because for years or decades, maybe even longer, we have insurance companies, and I think they contributed to the widespread distribution of standards and use and wide acceptance of the standards. This is not so much in veterinary medicine, and I think we need to start thinking, how do we apply standards? It's more challenging because we have lesser number of people, and we also have higher number of species that we are working with. But it's something that I think is it's maybe not a low-hanging fruit, but it's something that we need to tackle sooner or later.
Absolutely. Thank you, Zvonimir. We'll keep going for maybe another 6 minutes. What kind of partnerships and support would help you scale this tool more broadly across the livestock sector?
Yeah, well, I think we need to have so a couple of thoughts. One thought is that about molecular data. So I have two thoughts, actually. We need to start tackling how do we collaborate between different jurisdictions. Genomic data sharing, from my experience, is insufficient. We need to start thinking, how do we have something that is national level? And that needs to be supported. Like data already there, but we need to put some thought into how do we connect this data, and how do we have people who are going to work across different universities or research institutions, I don't care, in order to utilize that information, because that's one of the key pieces of information. So sequence information, widely abundant, available, but I have a feeling that we are not utilizing that properly. The other one is integration of this text analytics, because majority of the data in veterinary medicine, and I'm not talking only about laboratories, I'm also talking about clinics, I'm talking about the practice information management systems. A lot of that is coming in the form of text data. And in other jurisdictions, Australia, UK, for example, to some extent Europe in general, they do collect information from multiple entities, and whether these entities are clinics or these entities are laboratories, at the end of the day, it doesn't matter. What matters is how do we actually make this all work together for the benefits of the entire health information system. So what is actually needed? We need to have collaboration at the national level, and we need to have some sort of strong funding scheme, because it's all about people, really.
Absolutely. I hope that's the next step of your project. Maybe a couple more questions. What additional research or insights beyond disease monitoring and surveillance could these dashboards provide for our understanding of the disease being studied?
Yeah, so I mean, dashboards are not a solution for everything. I think we are battling a little bit with, of course, availability information, the accuracy information, but also privacy concerns, right? There are certain questions that will always be addressed by specific research projects, right? If you really want to understand how disease is behaving on a small scale, we need to have research projects. But I think, there is a certain level, I would call that a stack, right? We have, the world, we have, the institution, we have functions, but we need to know where to stop with these dashboards. Dashboards are not solution to everything, and even if there is additional layer of granularity and specificity, we cannot really go there, because A, we are missing data, and B, it could be it's not going to be aligned with privacy and confidentiality, and this is something that we don't want to do. For example, when we are doing spatial information, we ask veterinarians what would be the as not only veterinarians but also animal health experts, what would be the right level of spatial granulation. And although there were people who were saying, well, we need to have information about the exact farms, I think majority vote was not a vote, but feeling was that the county is as low as we can go, and some industries do not even want to have county, because that could be too spatially insensitive. So if you don't have really very, very specific granular information about what is going on in your barn or in your region, you can have a big perspective, but you cannot really address patient level or farm level recommendation, right? So there are limitations to what dashboards can actually do, for sure.
We're just approaching 2:50, a few minutes over our ending time. But thank you so much, Zvonimir, for answering all of our questions and sharing your insights. I did want to say we have a few more questions, but unfortunately we don't have enough time to answer all of them. So we will be sharing feedback from Zvonimir with those questions. As well, you can see for more information about the dashboards and the Food from Thought Agri-Food Pathogens report and the Industry Focus report, you can scan those QR codes for more information. And just a huge thank you to Dr. Zvonimir Poljak for your time and insight and all of your research work with Food from Thought. Thank you to Charlotte Wall, our IT help today. You've done an amazing job in the background. Thank you to LRIC and Food from Thought's collaboration on this webinar. And also thank you to our audience for attending. We really appreciate your time and learning more and being open to learning about the research from Food from Thought, and maybe forming amazing collaborations that can continue this very important and relevant work. This recording of the webinar will be shared on the LRIC Livestock Research Innovation Corporation website, as well as shared after with you who have registered to the webinar. With that, thank you so much, everyone, and I hope you have a wonderful day.